Hi, everyone. Welcome to this lesson on scientific method and other various topics that are tested on the HiSET science test. My name is Gail Gosh. My goal today is to present to you some information about the scientific method, also to give you some practice in how HiSET tests these questions. So I guess I'm going to stop sharing my own camera here, and I'm going to start sharing my PowerPoint. Okay, hopefully you should be able to view my slideshow. I'm going to go ahead and get started. Okay, what is the scientific method? The scientific method is the process for objectively establishing facts through testing and experimentation. There's different layouts or different steps that are outlined. You don't have to memorize these steps, you just kind of have to be aware of it. This particular outline shows 6 different steps. I've seen it as small as 4 and as many as 8. But in general, you are thinking about, you notice something, and you notice that when the temperature rises outside, the leaves begin to wilt on the plants. And so you wonder if there's something about the temperature and leaves wilting. And so you might want to research that topic, but you want to have some kind of a hypothesis that you're going to test. And then you're going to plan an experiment. You'll analyze the data and look back at the results and then report the conclusion. We don't ever make an observation and say, oh, that must be the connection there, that the temperature is rising outside. The ice cream sales in Florida are skyrocketing. It must be that high temperatures cause us all to go crazy and buy ice cream, or whatever it might be. You have to test that out and remove a lot of other factors. And so that process is what I'm going to be talking about today. First of all, the hypothesis must be testable. So I've got 2 questions here. Which species of flower is the most beautiful? Well, that's a matter of opinion. You can do a survey on it and report that, certain percentage of the population feels that, the peony flower is the most beautiful or whatever it might be. But that is not, fact. That simply because it's a question about an opinion. What species of wheat produces the most grain? Well, that can quantitatively be measured and answered. And so you might want to do an experiment, to just test the different species of grain as to which one provides or produces the most grain. Now, this is just a very simple diagram. The whole purpose here is just to point out a couple factors that are implicit in every experiment. An experiment must be well planned. I also teach the stats on the credit side of things, and so this is really right up my alley. But there's a couple of things that you need to be aware of: independent variable and dependent variable. The independent variable is the thing that you are manipulating, or that you are varying from one pot to the next on this one. A dependent variable is the outcome that you're trying to measure. So independent variable is the item that you are regulating or that you are manipulating. The dependent variable is the outcome. Okay, the experimental group and the control group. The experimental group is the group that gets the manipulated independent variable. The control group is the one that does not get anything. So the experimental group you're actually experimenting on. The control group you're just holding, you're safeguarding that just as something to compare to. Now, lurking variables must be identified and managed. You might want to, in this particular case, first diagram there, you can see we have 10 bean seeds per pot. They're each going to be planted in the ground in each pot and placed in front of a window. You can see here in the middle diagram, we've got the independent variable is the amount of water. One pot is going to get water, the other one is not going to get water. So the one that is getting the water is the experimental group. It's receiving that experimental variable. The control group is not. Okay, and then our last picture here is our dependent variable. This is what we are observing to measure an outcome on. So we are looking at the number of bean sprouts that sprouted over the time period. We've got 9 out of 10 bean sprouts sprouted in the pot that had water, 0 sprouted in the pot that did not have water. So that gives you a very, very quick, brief overview of the scientific method. Now, a lurking variable might be if I placed one pot in a east-facing window and another pot in the west-facing window, or if I had different potting soil in each of these pots. The result may be due to the amount of light and the type of light, bright morning sun or hot afternoon sun, or the soil. Did I have fertilized soil or not fertilized soil? Those would be lurking variables that would really impact your outcome if you are not measuring and managing those as well. So that's a big thing that students really need to be aware of, is lurking variables and other things that impact. Okay, so next thing I want to do is just go ahead and give you some examples with simple isolated questions that HiSET might present to you. And then I'm going to go to some more involved questions that you will definitely see on the HiSET as well. Okay, so in this first one, 2 students are planning a study to see which of 2 brands of vanilla yogurt is preferred by the students at their school. Which of the following is least important to do in the investigation? Let each participant taste both brands. Give the participants the yogurt without telling them which brand is which. Using only students who prefer vanilla yogurt to other flavors of yogurt, and make sure both brands are equally fresh. Now, again, we're looking at this and identifying which of the following is least important to do in the investigation. I would think that, obviously letting them taste both brands is going to be important. Giving the participants the yogurt without telling them which brand is which, I think would be important. If you told them, one was one brand and one was another brand, they might have preconceived notions that impact their choice. Use only students who preferred vanilla to other flavors of yogurt. I don't know that that's going to be preferable. The most important thing, make sure both brands are equally fresh. That's for sure needs to be. You can't have one that's nearly outdated and the other one being fresh. That could possibly impact your results. And so you could see that C would be the answer there. So I guess I'll go on to another one. A new soil additive is supposed to increase plant growth. To test this claim, students used 6 identical plants. Of these 6 plants, 3 received the additive and 3 did not. The table below shows the height in centimeters of the 6 plants after 4 weeks. So this first column is showing the height of the plant in centimeters for the 3 plants that received the additive. And the right column is showing the heights of the 3 plants after 4 weeks that did not receive the additive. Okay, so look at our outcome or our question here. How do the results relate to the contention that the new additive will increase plant growth? Well, contention is that hypothesis or that theory that you're testing here. So how do the results relate to the theory that new additive will increase the plant growth? They support it, but do not prove it, possibly. They prove it to be true, they disprove it, they cast doubt on it, but do not disprove it. First of all, being a stats person, I have to tell you that no experiment proves or disproves anything. It either supports the hypothesis or it doesn't support it. Because you might have an experiment that has some lurking factor that you didn't keep in mind, and somebody else might do that same experiment controlling that lurking variable and then still have other outcomes. And so all that you can say is that this experiment either supports or does not support. So right now, you don't say, they support it but do not prove it. That could be a possibility because they're clarifying that you don't prove it. They prove it to be true. They disprove it to be true. B and C, you can eliminate because we don't prove things. D, they cast doubt on it but do not disprove it. So we're here looking between A and D. Going back over here, generally what you would probably do is you would take these 3 plants and average out their heights. The same thing with the second column. But just looking at it with, just speculating here, looking at these 6 plants. I mean, the highest plant here was a plant that grew to 22.1, and they didn't get that plant didn't get the additive. All of them are between 20 to 22. Here again, we've got 20 to 21.4. I would say they cast out on it but they don't disprove it. In other words, there's no outcome here that you could say one way or the other. Outside of that, okay, which of the following observations would provide the best evidence to support the contention, or again, the theory, the hypothesis, that a certain substance causes high blood pressure? The substance is found in all people with high blood pressure. That's a possibility. It may be there, and that it's found in everyone, but it's there with people who have high blood pressure. The substance is rarely found in people who do not have high blood pressure. That could be important too. That could be it. The substance is chemically similar to another substance already known to cause high blood pressure. I don't know that could be. Carbon dioxide and carbon monoxide are two different gases that only differ by one molecule, but they have totally different impacts. So chemical similarity may not be what we're looking for. People who do not have high blood pressure develop high blood pressure when the substance is administered to them. That last statement reflects an experiment that you controlled a situation where people did not get this substance and others did, and the impact that it had to their high blood pressure. So it would be D. The others, you're just looking at an observation. An observation is never going to prove anything. You can say there's a similarity or there's a relationship there, but we can't say that it supports one thing or another. You have to experiment to identify if this is the key element that is causing that impact. Now, in all of these questions, I hope that you are seeing that you need to be able to read and make sense of it, that you're not going to be going back and identifying what was a lurking variable, what was the independent variable. You have to be able to make sense. So don't feel like you have to memorize much of anything. You have to be able to critically think about it. So that is really what I want, one of the things that I really want to get across today. So I had some other things here, but I'm going to go ahead right into some questions that we've had on HiSET in the past. So right now I'm sharing with you a display. It's a couple sample activities that I found in old HiSET qualifying tests that I thought really provide a good example here as to what's going on, what you might be tested on. So I just want you to see that both of these things are the same document side by side. I had to share my screen here and put it up here twice just so that I could get the actual information displayed here, as well as, sorry, I have a mouse that is going ballistic on me here. I won't move my mouse. Okay, so on the left side here, I'm giving you the information that you should read through and get comfortable with before you go anywhere near the questions. Now, I've got the answer key here just so that we can keep things moving. But I want to do kind of a think aloud activity here where I read and I process the information. I go through the questions just as I was before, helping you to identify what you could eliminate and what's most important and how to interpret. So we're just going to do some think aloud. Okay, background information here. Always take the time to read the background information and get comfortable with the information that they are displaying in any kind of a data display. So petroleum is formed by slow chemical changes in organic matter buried in layers of sediment in the Earth's crust. As the layers become progressively deeper and the pressure on them increases, petroleum is squeezed out and migrates upward. If the surrounding rock area layer is porous, such as sandstone or limestone, petroleum will collect in the pore spaces of the rock. So porous means spaces in the rock. If the rock is non-porous, such as granite or shale, the petroleum is trapped and will form pools below this rock layer. So non-porous is very solid rock, has no spaces in it. Demonstration. An earth science class performed a demonstration to observe how petroleum reacts when placed on various materials found in Earth's crust. 4 different materials were placed on newspapers on a lab table. Using an eyedropper, the students slowly placed several drops of cooking oil onto each of the materials. For each material, the reaction of the cooking oil was recorded. So this table is showing us what happened. So let's kind of get ourselves comfortable here. In our first column, we have the material. First one is a pile of sand. In the second column, we have the reaction of the cooking oil. So the pile of sand, the oil sank into the sand. For the flat piece of clay, it formed a pool on the clay. For flat rock 1, it penetrated the rock. Penetrated means it went into it. Flat rock 2, it formed a pool on the rock. So the questions are down here below, but I've put them over here to the right here so that you can see them more clearly and not swirling up and down on my display. In the demonstration, which of the following was purposefully changed so that its effects could be observed? They're inherently really asking you, what was the independent variable? What was manipulated and changed? But you have to understand what that meaning is. So we're thinking about, what did we change here? The amount of cooking oil, the reaction of the cooking oil, or the type of material from the Earth's crust, or the amount of pressure? Well, you can see the answer here is what they were varying was the type of material. All of these items here in the left column of our display were varied. Okay, based on the results in the table, which of the materials would be considered non-porous? So when I look over here, non-porous, I have to go back up here and remember, what did I say about non-porous? I realize my mouse is kind of all over the place, so I'm trying to point with it and it's not helping. But in this first paragraph, the last half of it, if the surrounding rock layer is porous, such as sandstone or limestone, the petroleum will collect in the pore spaces. So we're looking for something that could have been non-porous. Well, collecting would have meant that it would have penetrated or sank into it. So our clay and rock, we're looking for non-porous. Non-porous was trapped and will form pools below this. So it doesn't absorb. So it would be the piece of clay formed a pool on it and formed the flat rock 2, which formed a pool on the rock. So that's how we came up with clay and rock 2. Based on the information provided, flat rock 1 could have been which of the following rocks? Well, let's go back over here to our table. Flat rock 1 was the third item, and the oil penetrated the rock there. Well, going back up here, the sentence that I started to read earlier, if the surrounding rock layer is porous, such as sandstone or limestone, the petroleum will collect in the pore spaces of the rock. In other words, penetrate the rock. So it must be that that particular rock could be sandstone or limestone. Which of the following modifications would be easiest to implement to make the demonstration more realistic? Now, these first couple, the first two, are like almost impossible. Use the pressures under which petroleum forms in the crust. Well, those pressures are huge pressures. I mean, an entire mountain's worth of pressure weighing down on, the next lower crust layer is impossible to duplicate. Using materials from rock layers deep in the Earth's crust. Well, how deep are we going? Are we going, miles down, drilling down? That's very expensive and really not realistic. Using only rocks rather than including sand and clay. Well, sand and clay are a big part of our crust, the surface of our Earth. That doesn't make sense. Use petroleum oil instead of cooking oil. Well, petroleum oil is made from the oil that we find in the Earth, so that would make more sense. To make it more realistic, using petroleum oil would make sense to make that more realistic. Okay, going on to this next one. I'm going to go on to the questions here, but we have a lot of background information and a diagram and a display here to interpret. So take the time to read all of this. Okay, when pores in the leaves of a plant open to admit carbon dioxide for photosynthesis, water vapor inside the plant is unavoidably escapes. This loss of water vapor is called transpiration. Much of the water absorbed by a plant's roots is lost through transpiration from its leaves. The roots must absorb enough water to replace the water lost through transpiration, or the plant will not survive. Environmental factors such as light, temperature, and humidity can affect the rate of transpiration. So I guess I just kind of want to make this realistic for you. This is definitely why our plants really wilt in the summertime on a really hot, dry day. Um, you look outside and you've got to water those plants because those plants need water to soak into the roots and then up through the plant and out to the leaves in order to keep that plant functioning. Okay, so transpiration can be measured with an apparatus shown in figure 1. I want to make room for that. Once a plant is placed in the airtight chamber, a computer records temperature and humidity readings over a period of time. From these readings, the computer calculates the relative humidity and the rate of transpiration. Now remember, that rate of transpiration is the rate that the plant loses moisture through its leaves. Okay, so our experimental procedure. 2 scientists investigated whether light has an effect on transpiration. A bright light was placed above the chamber, and the top part of a well-watered bean plant was sealed in the chamber. Temperature and humidity readings were taken over a period of time. The chamber was then opened so that the humidity would drop to what it had been at the beginning of the investigation. The plant was resealed in the chamber, and a heavy black cloth was placed over the whole apparatus. Then a second set of temperature and humidity readings were taken. So the results, the transpiration rates, in other words, the milligrams of water lost per hour for both sets of data are graphed in the figure. So I am going to just take some time to look at this drawing and this display. So I've got my plant, I've got my chamber, I've got that little sensor in there with the plant that is sensing and recording the humidity and the temperature inside the chamber. The rest of this doesn't really matter that you have the support block, but you basically have the top part, the leafy part of the plant in this chamber. Okay, on the right side, we've got a data display. Now, usually your input, well, I won't go into that, but make sure that you understand what is being measured along each axis. So on this horizontal axis, we have decimals from 0.5 all the way up to 0.7. This is relative humidity. In other words, the percent of moisture in the air. So on a hot summer day in Iowa, very often it's very, very humid. In other words, you might hear the weatherman say, 88% humidity. Very, very sticky day because there's a lot of moisture in the air. So that's what they're displaying here, but it's in decimal form. Okay, along our horizontal axis, we have the transpiration rate. In other words, milligram per hour. This is the number of milligrams of moisture that is released into the chamber, milligrams per hour. So in other words, for this 4 milligrams, this rate, 4 milligrams of water are released into the chamber in 1 hour. At this rate, the highest rate, 32 milligrams released into the chamber in an hour. So make sure you get acquainted with all of that. Now I'm looking at our data points here are these circles and squares. The circles represent readings in the bright light. Okay, and we only have, I don't know why they only had 5 readings of that. And they have 8 readings where the plant was in the dark. Okay, these lines are something we call trend lines, and they're just there to identify the trend of the data. And you can see that as the relative humidity increases down here, goes from 50% all the way up to 70%, we can see that the amount of moisture lost in this plant falls in both cases, whether it's in the light or not in the light. Okay, so that's a linear trend there, and that's what that trend line is really trying to accentuate there. Okay, going on to our questions. A dependent variable in an experiment is the factor that is observed to see how it responds to a systematic change in another variable. What was the dependent variable in this experiment that they are trying to measure? They're not trying to measure the type of bean plant or the amount of light. The number of days in the chamber don't make sense, but the transpiration rate, that's what this chart is all about, is this rate of transpiration given the humidity and whether or not it's in the light or in the dark. So we're looking at C there. The main reason the chamber had to be airtight was to prevent the escape of which of the following? Heat, light, carbon dioxide, water vapor. Yes, it's trying to measure the humidity, which is the vapor, moisture in the air. So the water vapor. Okay, which of the following best describes what happened to the transpiration rate as the relative humidity increased? I just made that comment there, that we could see that as relative humidity increases, we can see that the transpiration rate falls or decreases. Okay, consider the two statements below. The plant lost water faster under bright light than it did in the dark. Yeah, the bright light, had higher rates of transpiration rate than in the dark. That's true. That makes sense. The transpiration rate is related to the relative humidity. Well, if it wasn't related, it would be the same measure all the way across, but we just commented that as the relative humidity increases, the transpiration rate decreases. So yeah, both of those are true. So which of the statements, if either, is supported by the data in figure 2? Both of these statements. Okay, which of the following is the best estimate of the transpiration rate when the plant is in the dark and the relative humidity is 60%? Now that's in decimal form. So I need to go over here and I need to look along my horizontal axis here and find 0.6. And then I'm going to look up to, I'm looking at when the plant is in the dark. So that is this first line here. And according to this trend line, that trend line is meant to give us an estimate, a way to estimate these points in between. So it looks to me like it'd be around 9 or 10 based on where that imaginary line from 0.6 up to that line is, the intersection there. Why was the black cloth placed over the apparatus? And the first answer there, I think you can see, to prevent light from entering the chamber in order to create these two different data sets here. And I will just let I'm just having a very hard time with the mouse here. Okay, when the relative humidity is 0.55, the transpiration rate of the plant under the bright light is about. So in other words, we're not looking at trying to find, a data value here along the y-axis here. This time we're trying to compare the plant under the bright light compared to under the dark light. So let me read the question again. When the relative humidity is 0.55, so I'm going to find 0.55 and count up. I just really need to get my mouse off the screen, and I'm hoping that you guys can look at the 0.55 yourselves here. Counting straight up, we can see that we'd land up here. When we follow that horizontal or that vertical line there, we could compare the reading when it's in the bright light compared to in the dark light. And we can see that it's about twice as much in the bright light as it is in the dark light. We went from like 10 up, 10 to 12, depending upon where our actual data point was and where our trend line is, all the way up to the data point in the bright light. It's about twice as much as when the plant was in the dark. Okay, so I'm hoping that you can see that you really have to be able to interpret the data. Very, very few times will you have a question like this one up here where you have to go to the data, you have to go to the display and read back and pull data off the display. It's generally you're going to have to interpret. So the best thing I can say to that is please take the time to read the information, analyze the diagrams and the displays. That's the best thing you can do to help yourself. So this is another one that's kind of complicated. So I will read this. A pendulum's period is the time it takes for the pendulum to swing all the way across and then back to its starting point. Students in one science class had observed that the shorter pendulums had shorter periods. They also noticed that if they blocked a pendulum in the middle of its swing, the pendulum would travel faster during the part of the swing in which the string was blocked. I guess I want to get you to think about a pendulum. An easy pendulum for you to imagine would be a yo-yo. You could, just let the string hang down, let the yo-yo hang, and, if you'd have it swing back and forth, you'd see that when it's a really wide swing, it's going to swing really fast. And when it's a more narrow swing, it actually slows down the actual period. That's what we're talking about. The time that it takes to get from one end to the other and back. That is your period, the time it takes for it to make one full swing. Okay, the pendulum, when it's blocked halfway along the string, the pendulum was then behaving as if it were shorter. The students hypothesized that the period of the pendulum blocked in the exact middle of its swing would be equal to the average of the periods for the full-length pendulum and for the half-length pendulum. That seemed reasonable because the blocked pendulum would be acting as if it were a full-length pendulum in the first half and as a half-length pendulum in the second half. And I think right now it might be a good idea to look at these diagrams. So a full-length pendulum means there's nothing blocking. And so measuring that period back and forth is what we're looking at. But if they blocked it halfway, they could see that this actually has the appearance of, moving faster, but actually has a shorter pendulum. It takes more time. And then here, when they blocked it halfway so that it's doing a full swing the first half and a blocked the second half, they wanted to see what the impact would be. So they cut 6 different lengths of string and hung a heavy lead weight at the end of the string. They then timed the periods while each string and weight was swung at a full-length pendulum, as a half-length pendulum, and then as a block pendulum. So that's what we are looking at here. And so this is our data. So I'm going to slide this so you can see the picture and our data. For each pendulum, the students used a stopwatch to measure the total for 30 complete back and forth cycles. The total number of seconds that was then divided by 30 to calculate the pendulum's period. Okay, so why did they not just do one swing? That's going to be one question there. But what they did, I want to make sure you understand this part. They took a yo-yo basically, and they let it swing 30 times, and they timed that for different lengths of strings. Okay, and different impacts based on, the pictures up here. They put it through each of those. Then they organized their data here. So we have 6 different string lengths, full lengths, and half lengths. Our middle 3 columns are that time that it took to go one full swing across and back. Now, remember, they actually calculated that by letting the pendulum swing 30 full swings, timed that full time, and then divided by 30 to get these measures here of what one swing, the time for one swing would have been. And then they took the average of periods full and half length, and they calculated that and added in this last column. Okay, so a lot going on here. What were the students trying to determine about a pendulum's period? Whether the period of the short pendulum is shorter than the period of the long pendulum. They had already previously observed that. Whether the period of the blocked pendulum is different from that of a free swinging one. Well, they knew that too. That's what they're checking out here, trying to really nail down what is that difference. Can we speculate a theory here? Whether the period of the block pendulum is more like that of a short or a long pendulum. They notice that it's not like either. It behaves differently. Whether the period of the block pendulum is the average of the periods of the related short and long pendulums. Now I'm going to go back here. This is their theory right here. The students hypothesized that the period of a pendulum blocked in the exact middle of its swing would be equal to the average of the periods for the full-length pendulum and for the half-length pendulums, like the middle sentence or third sentence of that first paragraph. Okay, so that would be D there. If a 55 centimeter pendulum unblocked, as in diagram 1, had been used, approximately what would have been its period? So I'd go back over here and I'm looking at the lengths of the strings and I'm like, wait a minute, we don't have a 55. Oh, that's right, we're speculating here. Well, the outcome should have been somewhere between 50 and 60. But which of these diagrams are we looking at here? It said in diagram 1. Well, we'd look at that first column in diagram 1 and halfway, 1.4 to 1.6, halfway in between there would be 1.5. So there, that's something called interpolation, where you don't actually have a data value for that input, but you can speculate based on the data that you do have. To see if the data support the hypothesis being tested, which two columns should be compared? So going back to here, they're looking at this third. I mean, they're very interested in how can we, what rule can we apply to this third column, this third scenario here? And they speculated that it was the average of the other two. Well, this last column is that average of C and D, or, diagrams 1 and 2. So we'd be comparing E and F. Okay, was it good experimental procedure to measure the total time for 30 cycles and then divide by 30 to calculate the period, rather than to measure the time for one cycle? Yeah, that's going to make, actually that's called duplication. The more you duplicate your experiment, the more confident you can be with the results. But yeah, one period is going to be too short to measure with any kind of precision using the student's procedure. I think the only way that you could, be more precise with just one experiment would be to kind of have a photo or camera finish like they do in horse races. But yes, for that question there. Okay, now back up to the other column here. What can be concluded about the hypothesis that the period of the block pendulum is the average of the periods of the related half-length and full-length pendulums? So I'm going to go back here. This speculation here, we're comparing columns E and F. We're speculating that they should be pretty equal. Well, this first trial, they were off by 1/10 of a second. Second trial, exactly the same. Third trial, it's off by 5/100ths, not even 1/10. The next one by 1/10, next exactly the same, and our last one off by 5/100ths. That's pretty darn close. Okay, it has been proven correct. Again, heads up, we don't prove anything. We either support or don't support the hypothesis. It is supported, but should be tested by further experiments. That is our answer, but I'm going to go on and talk about the others. It must be wrong because the data do not support it or fit it exactly. Again, there's going to be human error and just experimental error. You're never going to get an exact thing there, so don't always look for an exact match of the data. It must be right because it's so logical. Well, again, we don't want to justify the logic behind this. You have to have the data supporting it. So back to B. This supports it, but we're going to do further experiments to see if we can, really nail this down. Again, it's that duplication. When you're duplicating an experiment many, many times, you're much more and getting the same outcome, you can be more and more confident with your results. Okay, 2 characteristics of a pendulum are listed below. The period of a pendulum stays the same while the swings become narrower. Remember, as I commented about that yo-yo, that as the yo-yo, proceeds with its swinging back and forth, it's going to get narrower, but it will also be slower. So that period, that time that it takes to get from one side to the other and back, is going to be consistent. The period of a pendulum does not depend on the mass of the object attached to the end of it, to the weight of it. Okay, the students calculated the pendulum's period by timing 30 periods and dividing by 30, which the characteristics of either is supported in this procedure. The fact that it stays the same while its swings become more narrow, that's why they chose to do 30 and divide by 30 swing, to do 30 periods and divide by 30 to get their time, because of the statement in statement 1 there. Okay, 3 possible changes in the procedure of the experiment are listed below. Use the second hand of a watch rather than a stopwatch, timing several 30-cycle trials, I should say, and then changing the order. Well, changing the order isn't going to make any difference, and I don't think a second hand is going to be any more accurate than a stopwatch. But doing this experiment many more times, several more times, is going to add credence to your outcome. I just said that earlier. I kind of gave it away. Okay, I'm going to go on to the next one. I'm going to display the whole picture here. Okay, the tendency of a liquid to rise or fall in small tubes is called capillarity. A class was interested in identifying factors that affect capillarity The illustration shows one of its experiments. So we can see that we have two different liquids here, liquid X and liquid Y. We have different capillary tubes. These are these small tubes. Well, we have narrow, medium, and large sized tubes in each of these liquids. But look at our picture here. In liquid X, we can see that the liquid rises in each of these capillary tubes, where in liquid Y, the liquid falls. We can also see that the more narrow the tube, the more intense the impact. Because here, the narrower tube has the highest level of liquid in it, whereas over here on the Y, liquid Y, the narrow tube, it's really gone below the level of the liquid in the beaker. Okay, given the results below, which of the following factors affect the height of the liquids in the tube? And I think I just discussed quickly that both the type of liquid, we have different outcomes for liquid X than from liquid Y. So we have different impact. Here it rose up, here it fell down. And the diameter of the tube, it was more intense the more narrow the tube was. Okay, so we're going to say over here, liquid both 1 and 2. The level of liquid in a glass capillary tube rises when the liquid's molecules are attracted more to the wall of the tube than to each other. That's what happened in liquid X. In liquid Y, they were not attracted to the wall of the tube. They were more attracted to each other, so they pulled away from that wall. Okay, so we can see that liquid X and Y have different outcomes. So that's one of the things that you want to check here. It can't be, more to its own or more to the tube. It can't both have the same effect. So B and C we can eliminate. So it's between A and D. Liquid X more to the tube than its own molecules. Liquid Y more to its own molecules than to the tube. Okay, so we'll go on to the next one. Actually, this next one, I can just get it all on one screen. Okay, here you're just interpreting information that on a table here. The chart below shows which blood types may be safely received by persons with other blood types. Yes indicates a generally safe match. No, it's not a safe match. So our rows are going to indicate the donor's blood type. The columns are indicating the receiver's blood type, and they each have O, A, B, and AB. So, and then we're looking at the intersection of these two. A person with blood type A can, in general, safely receive blood. So I'm going to go up here to the receiver and columns, and I'm going to go to column A. They can receive, in general, from which of the following donors? Looks like O and A, and that's it. So O and A only. Okay, which of the following statements is not true? A person with blood type A can be a donor to people with type AB. So I'm going to donor and comparing to the receiver. Okay, so A to AB, yes, that's true, but they're asking for what's not true. Okay, so I'm looking for the intersection between donor, the row for donor A and the receiver AB, and it's a yes there, that intersection. A person with type O can be a donor, so I'm going to go to the top row. Look at that. They can be a donor for everybody. So yeah, that's true. I don't even have to specify whether it's A or B. A person with type AB blood can be a receiver, so they changed it up. AB, receiver, those are our columns. So I go to the AB column, can receive from type O, a donor type O. And that's a yes, that's that first cell in that column there. A person with type B blood can receive. So again, I'm looking at the B column here, and they can receive from type AB. I go down there, and that intersection says no. So that is our false statement. That's the statement that is not true. Okay, and this is our last one. I know you're relieved. Okay, you'll very often need to interpret different diagrams. This one is just an ecological pyramid. Basically, the premise on an ecological pyramid is that you have more organisms at the lowest level, and fewer as you go up, and that the upper ones are prey or are dependent upon everything below it. Okay. At which level of the pyramid are producers usually found? Well, plants are producers here, and they are going to be at that lowest level. Everything above that is going to be dependent upon plants. Okay, from the top to the bottom in the pyramid, the number of organisms in each level is expected to increase. Thus, we have the size of the pyramid. That lowest level has to be the largest, to be the foundation for a pyramid. But it kind of makes sense. That's where all the producers are. There's got to be way more producers. Heaven forbid if there were more wolves than there were plants, this ecological pyramid would be over with. Okay, and our last question here. In an ecological pyramid containing squirrels, nuts, foxes, and grizzly bears, which level would the squirrels be? Okay, so the plants are going to be at the lowest level. Up from that, you're going to have small animals that live entirely off the plants below it. Above that, you're going to have animals of prey that live off those small animals that are dependent upon the plants. And then above that, you have birds or animals of prey that are the highest level. There is no other animal that lives off that animal. So they are, at the top of the pyramid here. So we want to see which level would squirrels be found at. I would think that squirrels would be much like the rabbit. Squirrels live off those acorns and whatever they stash away for hibernation, but they live off plants. They don't live off or prey on other animals. So that would be most like the rabbit, level 2. Okay, so that ends my presentation here on different things to be aware of with science. I'm hoping that you can see that scientific method is, it's tested, but it's not really tested. They give you different experiments, and then you have to interpret. The other thing is there's lots of diagrams to interpret. Now, you are going to be given a link during this presentation to go in and practice yourself on a HiSET science practice test that was actually used probably 3 years ago as one of our qualifying tests. So it's a pretty recent rigor. It will give you a good indication as to how you would fare on a HiSET science test. It's also going to give us a good idea. So your proctor here is going to probably provide a link to that in the chat. I'm also going to ask that the proctor also provide a link to both my PowerPoint and the presentation that I just did here, these two, the answer key at least, if you wanted to take a look at those as well later. So you will have access to all of this. So that's what I have for science. I'll be presenting another one on social studies in just a bit.